Hello everyone, I welcome you to another video by Legacy IAS, a video which is going to talk about the future of how we can basically have diseases being treated, diseases as fatal as cancer being treated. Sometimes we can also maybe manipulate the genes of an upcoming child, all right, of a newborn child, of an unborn child. So there are a lot of such new revolutions which the world can basically see, and something that has made news in the recent days, which is the CRISPR technology or Cas9 technology, right? So in this particular video, which might be little technical and something that may require a lot of attention, a lot of detailing, we're going to try and understand and delve deep into this particular issue and what the technology is all about, what is the working of this particular technology, what are the benefits of it, and of course some issues which also revolve around it. So let us look at this entire discussion. Now firstly, we need to understand that what is this genome editing. All right, now genome editing is nothing but another name for gene editing, a group of technologies basically that gives the scientists the ability to change an organism's DNA. Now, this DNA, which basically determines the characteristic of an individual, all right, this is particularly so intrinsic and makes one individual very different from the other. Any alteration or changing in this particular DNA is basically called genome editing. So the technologies which are used to kind of bring about these particular changes. Now these technologies allow the genetic material to either be added or be removed or altered at particular locations. So you can actually make any of these alterations by addition, subtraction, or even altering this particular genome. This particular process is often used for the prevention and treatment of human diseases, including the single gene disorders which are like cystic fibrosis, hemophilia, single cell disease, and much more complex diseases like cancer, heart diseases, mental illness, and also HIV infection. So we're looking at a whole different gamut and whole new gamut of diseases which our current modern medicines have not been able to have a foolproof preventive plan for them, and we're trying to address it through genome editing. Now, as far as CRISPR technology is concerned, basically CRISPR or Cas9, which is in short for clustered, regularly interspaced, short palindromic repeats and CRISPR associated protein 9. This particular gene editing technology is something which received the Nobel Prize also in 2020. Now, how does this particular CRISPR 19 work? What are this entire mechanism and what do we get out of it? This is something which was adapted from a naturally occurring gene editing system, all right, whereby a bacteria is in use as an immune defense. So here the importance of bacteria is being mentioned in this particular technology and its entire functioning. So when infected with any virus, what this bacteria does, it captures the small pieces of this virus's DNA, the DNA of these particular virus, and inserts them into their own DNA, into the bacteria's DNA in a particular pattern in order to create some CRISPR arrays. Okay. Now, these CRISPR arrays in turn allow the bacteria to remember this particular virus whose DNA it has seen and looked at. Now, if the virus again attacks them, this particular bacteria, what it does, it produces RNA segments. All right, from these particular CRISPR arrays we just talked about, that recognizes and attaches to specific regions of that particular virus DNA. All right, so this particular part is very, very interesting, how the bacteria kind of produces the RNA segments and attaches it to that particular virus's DNA. Now, this bacteria then uses this Cas9 or similar enzyme to cut this DNA apart, which disables the virus. And the next step is basically editing that you use to alter that piece of DNA. So genome editing is there, but there's also the very important role which is being played by this particular bacteria in the journey of genome editing. Now, the benefits of this particular genome editing are actually a lot. It is not like this kind of a system of which is being done by CRISPR currently. It was not there earlier, but the extent to which it is fast as well as cheap, and the accuracy and the efficiency with which it does is something very, very unique and has been awarded with a Nobel Prize. That is why. So this particular technology has the potential to transform medicine like never before, enabling us not only to treat but also prevent a lot of diseases in this regard. Here we have the capability to even determine the genes of our unborn child. So there can be multiple uses that can come out of it. Some say super babies can basically come out in this scenario. The benefits also go to the extent that we can use it for fingerprinting the cells. These particular proteins can also be programmed to find and bind to almost any target sequence, all right, so that we can give a piece of RNA for its search. This particular CRISPR technology can also be used to make changes and replacing any kind of faulty genes which are basically there and ensure and bring in the genome editing. The customized Cas proteins can also be created which basically do not cut the DNA or even alter in any way. It only kind of turns on a particular gene and turns it off, which is basically called as CRISPR A or CRISPR I respectively, if you are turning it on or if you're turning it off. So here the base editors, by changing one letter of the DNA code to the other, can basically perform the function of altering the entire DNA. Now the research team basically developed a new approach whereby we are trying to decipher the changes which are coming in the genome as so that we are giving a much more targeted locus to the effects that can take place in this particular genome. Now, this particular technology is also used in humans to remove certain specific genes so that the immune system can be activated against various fatal diseases like cancer. In fact, we have someone from the University of California commenting that this is a leap forward in developing a much more personalized treatment to cancer, whereby the isolation of immune receptors that specifically recognize mutations in the patient's own cancer are used to treat the cancer, right? So the way in which we have basically the mutations in the patient's own cancer trying to use and treat the cancer is trying to give a kind of personalized treatment for cancer, and that's very novel. In fact, we are also using in India for a lot of temperature sensitive organism, plants and crop varieties, and bring in the necessary gene edits which are required. So the benefits that we're looking are absolutely enormous and amazing, but of course there are also some issues involved in the entire process. Firstly and very importantly, the ethical concerns, the way in which you can actually trying to alter the entire human genome. Now, most of the changes that are basically introduced are limited to the somatic cells. The germline cells and the genome editing bring up a lot of ethical challenges. The extent to which you can basically alter the human traits, the ethical aspect of it is something which is being highlighted and being talked about, that to what extent can you change, let's say, someone's height or someone's intelligence. To what extent can we allow technology to have this kind of control? So based on the concerns about ethics and safety, the germline cells and also the embryo genome are currently illegal in several countries, including the United States. There had been an attempt to change the genomes of children in China, but that was considered as premature and also unethical. So they thought that it could probably benefit children, but it was treated as unethical. In fact, there have been some efforts whereby schools have also tried to have experiments as to how they can kind of have this genome editing in United States. Some experiments have gone about. Now the point here that we have to note that firstly, this technology technology can be utilized, and if the way in which through bacteria, if it's really become successful, it is capable of bringing enormous amount of changes and cure diseases like cancer and HIV. But at the same time, the ethical concerns, which means that it has to be done sustainably, all right, so that we don't really allow technology to overpower the way in which humans kind of lead their natural life. This kind of dilemma is where we are at this particular technology in today's time. I hope you have understood well from this particular discussion. Let me know by liking that button. Share it with those who still think that they cannot determine the genes of their unborn child, and subscribe for more such videos from Legacy IAS.